All right, so we're diving into orphan receptors today.
Oh, yeah. This is a fascinating area.
You sent over a ton of papers
Yeah.
and even a YouTube lecture on this.
We
So
had to make sure you were prepared.
yeah, you really went all out. So we're definitely going deep on this.
For sure.
We're talking about these mysterious receptors, the ones that we don't know what activates them.
Yeah.
Like a lock without a key.
Exactly.
And that's the exciting part, figuring out.
Yeah, how do we find these ligands, these keys?
Yeah, and what do they unlock in terms of new therapies? That's really what we're looking at.
Yeah, so many potential targets out there.
Cons.
If we think about it, we've got all of these receptors, these locks in our cells,
Yeah,
just waiting for the right key to come along and change how our bodies function.
absolutely.
I like that analogy.
Yeah.
So before we get too far ahead, let's start with a classic example.
Okay.
Thyroid hormone receptors.
Sure.
They were some of the first receptors discovered,
Right.
but their story's pretty complex.
Oh, yeah, absolutely. So thyroid hormone receptors, or TRs,
Mm-hmm.
they're kind of like a master key
Yeah,
that opens doors all over the body,
that's a good way to put it.
involved in everything from brain development and bone growth
Mm-hmm.
to even the transformation of a tadpole into a frog.
It's true. Metamorphosis.
No way thyroid hormones control that.
It's amazing.
That's crazy.
Yeah.
These receptors are clearly super essential for life.
Oh, yeah, for sure.
But what happens if they don't have their key, their ligand?
That is when things get really interesting.
Okay, I'm intrigued.
Because you see, unligated thyroid hormone receptors can actually silence gene expression. Like they have a built-in mute button.
Really?
Yeah. So when that key, the thyroid hormone, isn't around the receptor,
Uh-huh.
it binds to a corepressor, a protein that blocks gene activity.
Interesting.
But then when the hormone does arrive,
Okay.
it kicks out that corepressor and lets a coactivator bind, which switches the gene on.
So it's like this little cellular dance party going on where the receptors, the dance floor,
Mm-hmm.
the hormones, the VIP guest,
Yes.
and the corepressors and coactivators are battling it out for a chance to dance.
That's a great analogy.
They try.
It really captures that dynamic interplay
Right.
between these molecules.
For sure.
And there's a detail here you shouldn't miss.
Okay, tell me more.
This part of the receptor called helix 12.
Helix 12.
Yeah, it's crucial.
Okay.
For this whole process, imagine helix 12
Uh-huh.
as the lock's keyhole.
Okay, I'm picturing it.
If it's the wrong shape, that key won't fit,
Yeah.
and the whole dance party falls apart.
Wow, so it all comes down to shape.
In a way, yeah.
That's fascinating.
And mutations in helix 12 can actually cause
Whoa.
resistance to thyroid hormone,
Yeah,
a condition where the body doesn't respond properly to thyroid hormone.
even when it's present.
Exactly.
Wow, so this tiny little helix has a massive impact
It really does.
on our health.
Huge consequences.
So we've been talking about TRs, which have a known ligand. But what about all those other receptors out there? The true orphans.
The mystery.
How do scientists even begin to find those missing keys?
That's the challenge.
Right.
Deorphanizing receptors.
Yeah.
It's like solving this complex puzzle.
For sure.
They use all these techniques like high-throughput screening,
Okay.
where they test thousands, even millions of potential keys
Wow.
to see if any of them fit the lock of that receptor.
So it's like trying to find the right key for a door
Yeah.
by trying every key on your keychain and then some?
Exactly.
That sounds pretty tedious.
It can be.
But I guess with today's technology, they can automate a lot of that.
Oh, absolutely. Yeah, they've gotten a lot more sophisticated
I can imagine.
than just that brute force testing.
Right.
Structure-based design is another approach.
Okay, what is that?
So they use the 3D structure of the receptor,
Uh-huh.
essentially like a blueprint of the lock,
Oh, wow.
to design potential ligands that might fit.
Interesting. So it's like this combination of old-fashioned detective work
Yeah.
and cutting-edge technology.
Exactly.
But I imagine there's a catch.
There's always a catch.
Finding a key that only fits the specific lock you're interested in.
Yes, that's the key.
Otherwise, you could be opening a bunch of doors you don't want to,
That's right.
leading to all sorts of side effects.
Unwanted side effects, yeah.
Exactly. So
Mhm.
specificity is crucial
It's crucial.
when it comes to drug development.
Absolutely.
Researchers are constantly striving for that,
Yes.
to find ligands that are highly selective for their target receptor,
Yes.
minimize the risk of those off-target effects.
Exactly.
So it's like designing a key
Yeah.
that only opens one specific door
Right.
in this giant mansion.
A very specific door.
So we've got these orphan receptors, these mysterious locks.
Yeah,
Scientists are on this quest to find their missing keys.
that's right.
But what happens when they do?
That's the exciting part.
Right. Do they always find what they expect? Or are there surprises along the way?
There are often surprises. Sometimes deorphanized receptors
Yeah.
reveal functions that nobody could have predicted.
Oh, really? Like what?
Like, take glycine receptors in the spinal cord, for example.
Oh, yeah. I remember those from bio.
You do.
They help control muscle movement, right?
That's right. They're known for their role in inhibiting motor neurons,
Uh-huh.
which prevents muscles from becoming overexcited and spasming.
Right.
But imagine the possibilities if we discovered entirely new functions.
It's like realizing your trusty old house key can also open a secret passageway
Exactly.
that you never knew existed.
It's exciting.
And speaking of glycine receptors,
Yeah.
can you remind me what glycine actually does in the spinal cord?
Oh, sure.
I feel like a little refresher would be helpful
Yeah.
before we go any further.
So glycine's actually a simple amino acid,
Okay.
but in the spinal cord, it acts as an inhibitory neurotransmitter,
Uh-huh.
essentially acts as the brakes on a car,
Okay.
preventing those motor neurons from going too fast and causing uncontrolled contractions.
So even a seemingly simple molecule
Yeah,
can have a complex role in our bodies.
absolutely.
And finding new functions for receptors like these could open up
Oh, yeah?
incredible possibilities for treating all sorts of conditions.
Absolutely. Deorphanizing receptors holds immense potential
Yeah.
for developing these novel therapies.
It's like discovering a whole new set of tools
Yeah.
for fixing things that were previously considered undruggable.
It's exciting stuff.
This is incredible. It sounds like we're just scratching the surface
Oh, yeah.
of what's possible with orphan receptors.
We really are.
But before we get too carried away with all this potential,
Sure.
let's take a closer look at some specific examples
Okay.
of orphan receptors and their newly discovered ligands.
Let's do it.
I'm ready to hear about some real-world breakthroughs.
Perfect. Let's start with G protein-coupled receptors, or GPCRs.
Okay.
These are the largest family of cell surface receptors in mammals
Wow.
and a major target for drug development.
Okay, I'm all ears. Let's unravel some of these GPCR mysteries.
All right. GPCRs are fascinating. They're involved in so many processes in the body.
Yeah, like what? Give me some examples.
Everything from how we see and smell to regulating our heart rate and immune responses.
Okay, so when we talk about deorphanized GPCRs,
Yeah.
what are some of the unexpected functions?
Well, one really intriguing example is the GPR17 receptor.
Okay.
Originally an orphan, but scientists found that it's activated by two different types of signaling molecules:
What are those?
uracil nucleotides and cysteinoleucotrienes.
Hold on, let's break that down a bit.
Okay.
What are those molecules, and why is it so interesting that they both activate the same receptor?
So uracil nucleotides are involved in a lot of essential cellular processes, like energy metabolism and building RNA, which is crucial for making proteins.
Okay, so like basic cellular housekeeping.
Exactly. And then on the other hand, you have cysteinoleucotrienes.
Right.
These are involved in inflammation,
Right.
particularly in allergic reactions and asthma.
So one molecule involved in basic cellular processes, and another that's a major player in inflammation,
Right.
both activating the same receptor.
It seems like a strange pairing.
It does. What's the significance of that?
Well, it suggests that there might be a deeper connection
Okay.
between inflammation and cellular metabolism.
Interesting.
Maybe they're not such separate worlds after all.
So by deorphanizing GPR17, researchers stumbled onto this potential link
Exactly.
between two seemingly unrelated processes.
It's a pretty big deal.
Yeah, that is a pretty big deal. Are there other examples where deorphanized GPCRs have led to these kinds of insights?
Absolutely. Another great example is the GPR35 receptor.
Okay, so another orphan receptor, another newly discovered ligand.
Yes.
What's so special about this one, and what did this finding reveal?
So this one's activated by kynurenic acid,
Kynurenic acid.
a metabolite of the essential amino acid tryptophan.
Okay, and what's so special about kynurenic acid?
Well, it acts as an antagonist
Okay.
at certain glutamate receptors in the brain.
What does that mean?
It blocks their activity.
Okay.
And glutamate is a major excitatory neurotransmitter.
So it helps keep the brain's activity in check.
Exactly.
Interesting. So by activating GPR35, kynurenic acid might have an even bigger impact on brain function.
That's what researchers are thinking.
So does this have any implications for treating neurological conditions?
That's exactly what they're exploring.
Oh, okay.
Because GPR35 is activated by kynurenic acid, it's become a hot target for developing drugs
Wow.
for conditions like schizophrenia and Alzheimer's disease.
Where glutamate signaling is thought to be out of whack.
Right.
Wow, these deorphanized receptors are truly opening up
They are.
new possibilities in medicine. It's incredible how much we still don't know
So much we don't know.
about how our bodies work. But I'm curious, how do scientists actually pinpoint these ligands for orphan receptors?
It can be a really tough task.
Yeah, it seems like finding a needle in a haystack.
It is, but scientists have some clever techniques.
Okay, like what?
One approach is reverse pharmacology,
Reverse pharmacology.
where they start with the known ligand
Oh.
and then screen a library of orphan receptors to see if any of them bind to it.
So instead of searching for a key to fit a specific lock,
Yeah,
they're testing a key on a bunch of locks to see if any of them open.
that's a good way to put it.
That's pretty ingenious. Are there any other methods?
Another powerful technique is expression cloning,
Expression cloning.
where they introduce a library of orphan receptor genes into cells
Uh-huh.
and then expose those cells to potential ligands.
Okay.
And if a cell expresses a receptor that binds to the ligand,
Yeah.
it triggers a signal.
And they can detect it.
Exactly, allowing them to identify that receptor-ligand pair.
It's like setting a trap for the ligand using the receptor as bait.
Yeah, it's clever.
That's pretty cool.
Yeah.
I imagine this requires some pretty advanced genetic engineering.
It does.
So these methods, along with high-throughput screening and structure-based design that we talked about earlier,
Right.
have been really instrumental in deorphanizing a large number of receptors. So it's a combination of innovative thinking and cutting-edge technology
Yes,
that's driving progress in this field.
absolutely.
And with each new deorphanized receptor,
Yeah,
we gain a deeper understanding of our bodies,
for sure.
unlock potential new avenues for treating diseases.
So exciting.
This is fascinating stuff, but I have a feeling there's even more to this story.
There is.
What other unexpected findings have emerged from this world of orphan receptor research?
Well, one particularly intriguing area
Yeah.
involves a class of orphan receptors called trace amine-associated receptors, or TARs.
TARs? Those sound mysterious. Tell me more. TARs, huh? They sound like something out of a sci-fi movie.
Right.
So what's the story with these receptors?
Well, they were initially thought to be activated by trace amines,
Okay.
which are found in really small amounts in our bodies.
Okay.
But as scientists dug deeper,
Yeah.
they discovered that TARs can actually be activated by a surprisingly wide range of compounds.
What do you mean?
We're talking about certain drugs, things we eat. A real mixed bag.
Wow, so these terrors are like multi-purpose sensors,
That's a good way to put it.
picking up signals from all over the place.
Exactly.
What does that tell us about their role?
Well, it hints that they might be involved in a much broader range of functions than we initially thought.
Like what?
They could be playing a role in regulating our mood, our behavior,
Interesting.
how we respond to medication, even the types of food we crave.
That's pretty incredible.
It is.
So by studying these orphan receptors, we're not just learning about individual molecules.
Right.
We're getting a glimpse into this intricate web of interactions that affect our overall health and well-being.
Absolutely.
It really underscores the importance of basic research
Yeah,
and how it can lead to unexpected discoveries.
for sure. And speaking of unexpected discoveries,
Well, let's hear more.
there's another fascinating area of research
Okay.
that focuses on their potential role in the gut microbiome.
Wait, the gut microbiome, you mean all those trillions of bacteria
Yes.
that live in our intestines? What do orphan receptors have to do with that?
Well, it turns out some orphan receptors are actually found in the cells lining our gut.
Really?
And get this, they might be activated by molecules produced by the bacteria that live there.
You're telling me our gut bacteria could be communicating with our bodies
It's possible.
through these orphan receptors?
Yeah.
That's mind-blowing. What are the implications of that?
I know, it's really exciting. It opens up this whole new avenue for understanding the complex relationship
Okay.
between our gut microbiome and our health.
Right.
It suggests that these bacteria might be influencing our bodies in ways we never imagined,
Like what?
impacting our immune system, our metabolism, even how our brains function.
This is amazing. We started by talking about orphan receptors as locks without keys, and now we're uncovering this vast communication network
It is pretty vast.
between our bodies and the trillions of microorganisms living inside us.
It's incredible, isn't it?
It's like we've stumbled upon a secret language.
That's a great way to put it. And the more we learn about this language,
Yeah.
the more we realize how much we still have to uncover.
For sure.
One thing is clear, though.
Look at.
Orphan receptor research is leading to groundbreaking discoveries with the potential to really revolutionize how we think about human health and disease.
So what does all this mean for our listener?
Well, by understanding these complexities of orphan receptors
Right.
and how scientists are working to deorphanize them, you're getting a front row seat to a cutting-edge field
I like it.
that's really poised to transform medicine.
It's exciting stuff.
It is.
as researchers continue to unlock the secrets of these intriguing proteins. Who knows what groundbreaking therapies and life-changing insights await?
The possibilities are endless.
It really is a journey of discovery, and you're now a part of the conversation. But here's a final thought to ponder.
Okay.
What if some of these orphan receptors aren't meant to be activated by just one ligand? What if they're designed to integrate signals from multiple sources, giving us a more holistic picture of our internal and external environment?
That's a great question to leave our listeners with.
It is something to think about that will likely keep scientists busy for years to come.
Definitely.
And maybe, just maybe, you, our listener, will be inspired to join them in unraveling the mysteries of these enigmatic proteins.